welcome to a new episode of the Computomics podcast. Our guest today is the co-founder and CEO of AgTech startup GeneNeer, Kinneret Shefer.
Hi, Anna. Thank you. Nice to be here.
We love to have you, Kinneret. I hear you're quite the force of nature, quite active, and a bundle of energy. So I wondered, maybe you can tell us, what do you do to relax? What's your tip for relaxing?
Well, I think that's a very important thing for a CEO because it is a very demanding position. So I exercise pretty much every day. I do yoga, meditation, stretch, power, aerobic. Each day something a little bit different, but I start with meditation and stretch pretty much every day. So yes, I take an hour for myself in order to start the day in a very relaxing state of mind.
That's wonderful, because I assume as a CEO, the rest of the day is intense and not as relaxing, but still fun. Can you tell us, how did GeneNeer come into being? What's the story of GeneNeer's inception?
So the way it started is that I originally are a hardcore RNA researcher. I finished my PhD at the Hebrew University, the genetic department. I investigated telomerase RNA and decipher the structure of the core of the complex, and received a Fulbright fellowship from the USA Embassy and continued to postdoc at the RNA Center at UCSC. So I'm a hardcore RNA person. But having all that knowledge, I wanted to make something out of it, something applicable, something that make a difference.
Mm-hmm.
I didn't want to continue basic research. I wanted applicable research. So I actually did ask my sister, she's a veterinarian, if she can help me, see my way. And she introduced me to an American business guy, and I gave him a lecture about RNA for an hour because I was very at the beginning of my way.
Mm-hmm.
And he was very polite. So he sat there for an hour and listened. And then he said, I have only one question. And I said, I was thinking, wow, he's really smart. He got all those structures and elements. Wow. And he said, my question is, okay, how do you make money out of it? What is the one thing? How do you turn it into something that someone will want to buy?
Mm-hmm.
And I always tell that story because that's a shifting point from being a research that wants to discover structures and function for your own curiosity to contribute to the science in general, to become someone who constantly think about how to make it something that someone needs. The industry, what is the industry? What is the market? What is the worth? How do I take my technology into something meaningful and applicable? Mm-hmm. And that's a shifting point because I did not expect that question. And it kind of
why would you as a researcher, right?
Yes, it switched something in my brain. And from there on, I can see that it's a long process that constantly move, my brain constantly move from research to applicability for needs, for market, for targets. This is one of I'm a person that likes to learn all the time. It's in my nature. But I took it to a different direction. Instead of continue doing more studying of science, I started to learn and study market needs and targets and where is my strength, how can I be different and really produce something that someone needs. And since then, it's getting sharper and sharper. And that was a few years ago, quite a lot of years ago. I think that today I deal very little with the science, and I can say that 90% of my time is investigated in where is the company should go, who are our partners, what are the market needs, etc. So it is a shifting point that I try to, create value for the industry and for the investors that invest in us. So, and it goes together. Once you become meaningful for the market and you generate something that is needed, then you generate value for the company and for the people that invested in it.
Makes total sense. I think thinking of what you're actually producing that is needed in the market. You were quoted in a, I think, 2024 article in CTech as saying, "quote I want GeneNeer to be the Pfizer of crop seed innovation." What do you mean by that?
There are many needs at the crop market, and the biggest challenge is time and money. And it's almost equivalent. It's almost the same. Time costs
Mm-hmm.
money, right? And when you want to generate a new trait in crop, it can take years. If you use breeding, it can take 15 years. It becomes very expensive. But there are many needs, and especially under the climate change, new pests arise, new challenges arise, drought tolerance is needed, nematode resistance is needed. Many traits are become very valuable,
Mm-hmm.
valuable. And I aim to develop trade in a year. So
A
a year
year.
and a half, a year and a half. It depends what trade it is. But from the lab perspective, from bringing something out, it still requires field trial and greenhouse trial. But from a lab perspective, from GeneNeer perspective, we can generate a trade in a year or a year and a half. And that is really fast. And I have a confession to make. Originally, originally I'm not a plant person. I come from mammals.
Ah.
I'm RNA, but mostly in mammals. And when I started to work on plant, it drove me crazy because it took so long. Even if you work on a very simple plant like Arabidopsis, it can take months. And I told my team, no, I'm not going to wait. Okay. Because with mammals, you make an essay and next day you have a result. And then you look at the result, you conclude what you want, and then you make another essay. And next day you can conclude another. So you can move relatively fast when you work on cell culture. But when you work on plant, you put them in the greenhouse and now you have to wait for them to grow in order to conclude something. And it was really unacceptable for me.
Mm.
So at the beginning, I thought it's a disadvantage that I'm not a plant person. I did take some courses, read the plant biology book, and etc. But I'm not a hardcore plant researcher.
Mm-hmm.
So I shifted the entire way of working. We hardly do anything in the greenhouse. Everything is being done in the tissue culture because there I can do things fast at a short time and get results in a matter of a week, which is in plant considered to be fast. But also I can work on parallel on different ways of getting my target. And
Mm-hmm. You mean like almost like A/B testing? You would have like different tissue samples and then do different things to them? Or can you take us
yes,
into the detail level a little bit?
yes. So I'll give you an example. We developed the superline. Okay, the
Mm-hmm.
superline is a platform for gene editing, which can extract itself out of the genome without the need for crossing or single cell assays. I just had a conversation on Friday, actually, with an American researcher, and he told me that to regenerate a protoplast can take between 6 to 12 months.
Mm-hmm.
Okay, that is a long time.
And for our audience, you should have seen the eye roll when Kinneret said long time. So I can feel your impatience through the
Yes,
screen as we
oh my
talk.
God, I'm not gonna do that. I'm not gonna wait that long.
Mm-hmm.
And so the Superline is actually gives you a platform when you, in a matter of a month, can generate different traits. And the beauty of it is that you can always go back to the source because it's tissue culture. So we grow them in tissue culture, we do the manipulation, we propagate them, and now we have this gene editing platform in. We can introduce different guide. We can do stocking traits. We can generate in parallel
Mm-hmm.
many genes in parallel, and then check which one is the most efficient. So by doing that, and we can always go back to the source, to the superline itself, and introduce another silencing element. So by doing that, we turning the trait generation into a kind of a very organized and very fast process that
Mm.
can always be controlled. You can always go back and forth. If you don't like the result, you don't need to regenerate it. It's in the tissue culture. It's you can propagate it, and you always have your backup. So it's a little bit like
Mm-hmm.
computer, you have the backup, do your whatever
The
program
editing.
you want. You don't like what you have, you can go back, you can add, you can delete. So it all the time, it's like a very efficient process.
Mm-hmm.
So that makes it very fast. Once you have the super line, you can now generate many traits. And why Pfizer? Because you probably remember the corona, and the corona
It
is,
does stick in the memory.
you still remember it, huh? They say there's going to be another one soon. Hopefully not. But the virus changed dramatically, constantly. And Pfizer had to constantly adjust their vaccine to the virus.
Mm-hmm.
GeneNeer developed a platform that can constantly fast adjust to the market need. If there is a new virus in banana right now that put bananas into reduce the yield in a significant way that can increase the prices and can be dangerous for the market.
Mm-hmm.
GeneNeer can address that in a matter of a year and a half, and in the crop field, that is very fast.
Mm.
So the ability to have those fast systems is very essential. And of course, it reduces the price, because if you can generate something in a year instead of 15 years with expert, yes, we do have a team of experts, but it's still a year and not 15 years. So I kind of really fast noticed that what really hard for me to accept is the time to market.
Mm.
So I worked on that, and we constantly putting that in our top priority.
Mm-hmm. I think that those numbers speak for themselves almost, like 1 year or year and a half versus 15 years. That's already a huge, 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 huge difference. I think you've also mentioned a few other things that make your gene editing technology maybe different from others around. Besides the difference in time and the fact that you work with cell cultures.
Culture in plants is tissue culture.
Sorry, tissue cultures. I see. I'm thinking mammals too,
I moved.
maybe.
I kept the greenhouse, so my R&D will be happy, because they like, they are plant people, so they must have a greenhouse, and you do have to have a greenhouse at the end.
Right.
But 95% of the work is being done at the tissue culture,
In tissue culture.
which is fast, which is fast.
Interesting. So, can I ask, like, from a customer or like project partner perspective, how would that work? Like, how do you work together with your customers?
So our business model, and I will refer to your previous question. It's true, it's not our only platform, and we'll talk about the other platform. And the other platform is very relevant to AI, so we'll talk about it in a little while. As for our business model, it has two verticals. One, we call it trade by design, where we collaborate with major food processors or growers, where they have a specific demand. They want the specific variety. I'll give you an example, a variety that is great, increase the yield in 50%, but has a specific problem that they cannot use it in the industry. That problem can be addressed using our tools. And the collaboration is that we being paid for the work that we do, for milestones, for success fee, and royalties. And we usually share the IP. So someone that has a need in the industry, drought tolerance for potatoes in Brazil, or whatever example you can think about, tomatoes, problems with broom rape in California, all kind of problems that are specific for a growers or a company or a processor. We can collaborate and address their challenges. That's one vertical. The second vertical is that GeneNeer knows exactly where the power is, what our technology can be due to the market. And we are focusing on a specific product development, which I cannot elaborate right now. That
A
is
teaser.
Yeah, that is we're not gonna commercialize. It's always a B2B business, so we're not gonna commercialize. We already have someone who's interested on that project. But that is bringing something all the way to validation, not just development. And developing such a product that has the potential to revolutionize the entire market is expected to be very valuable. And that another advantage, guess what? It's pretty fast.
I see a theme running through.
Yeah. So yeah, you can understand how frustrated I was at the beginning. Right
Mm.
now, that's my goal, save time.
Mhm.
So this product is expected to have very high billions of valuable, but still get to the market in a reasonable time frame. And we are very exciting out of it, but we're keeping it confidential for very obvious reasons.
Of course, I'm sure we'll hear about it when it's ready to be shared. I wonder. I mean, if we think of gene editing technology, at least in Europe, there's often safety is often a very prominent topic when it comes to discussing that. What would you say needs to be maybe made safer, or potentially does your technology make safer when it comes to the challenges in gene editing?
So gene editing does not add any foreign DNA into the crop. So it's not a GMO product.
Mhm.
It's a gene edited product. It means that we basically speed evolution. Again, time. We speed evolution. Instead of waiting for something to happen randomly or by 15 years of selections, we are introducing and making small changes, basically usually deletions, into small deletion. It can be even one nucleotide. It can happen randomly. It's just going to take a long time.
Mm.
So we are encouraging it. We are speeding the evolution in a way and using CRISPR basically to generate that change. But at the end product, you have no foreign DNA. And I think in that sense, it's simply not dangerous because that crop could happen and that change could happen and did happen in some cases randomly. So from a regulatory perspective, I think that 99% of the researchers do not see any challenge or problem or safety issues. I think in Europe, if I may be very straight, it's more a political thing, not a
Mm.
science thing.
Mm.
You can see that the entire world is moving towards, and I think that Europe will eventually. You can see that it's open up in the US, in Canada, in Brazil, in Australia, in Japan. Everyone look into it and understood that this is a safe and very promising. And if you look at the prediction, the growth rate, it is expected to be above 70% rate. And it is expected to be the technology that will change the crop yield in the next decade. So all
Mm.
the parameter shows that this is the future. And I think that Europe has to resolve some political issues, but once they will, that will be out of the table and it will be more free market.
Mhm. Yeah, I would agree. It's there seems to be such an overwhelming opinion as far as the scientific community is concerned, but there's still a political reality, and the people who vote who also need to understand how it works. So I think it's important to make that distinction between the GMO and the gene edited products.
It's more than that. Right now, it's pretty much they voted for, they approved it, but now it's
Mhm.
like more who's going to own the IP and stuff like this.
Mm.
So it's not about the technology itself. It's just a bureaucrat and politics. And I think it will be resolved in my perspective, right? I think it will be solved.
Mhm.
But I do want to talk about the second platform that we
I was
developed.
gonna say maybe we can do that. Time is running short always when the conversation is this exciting. But yes, please do share what is the AI product that you have in line.
AI is a tool that helps us in many aspects, but I'll try to show it in one perspective because we do use
Mhm.
it. And the Mirtron platform, it's another platform, not the Superline. The Mirtrons allow us to tissue specific, time specific, silence and express a specific trait. So for example, some traits you want them to be expressed only during flowering. You don't want to
Mhm.
have them expressed or during other times, or even during a specific tissue, like at
Mhm.
the root. Or sometimes you even want to control the magnitude of the trait. You don't want to have all or none. That is what we call the next generation of gene editing. It's a more precise. And by doing that, we are making sure that no other parameters are being disrupted, like yield.
Mhm.
I'll give you an example, and I'll explain to you why is it AI, involved in AI. So we can choose every gene, and we need to use computer tools to select the right genes that express, let's say, only at the root.
Mm-hmm.
And then we can design the mirtron, but then we have to replace an intron with a mirtron. And we have to use AI to choose the right introns that will not introduce new sequence, because the gene editing is based on not introducing new sequences
Mm-hmm.
to the genome. So we are using AI in order to select the right gene that will be expressed only at the root. And let's say that we know such a gene in tomatoes, but maybe we do not know what the homologue in potatoes.
Mm-hmm.
So, and we do want to do it at potatoes. So we can use AI tools to predict what will be the gene that is behaving in a certain way in tomatoes that will be homologue in potatoes. What will be the right intron to target that will be expressed in the way that we intended to be expressed in order to silence and to generate the new trait. So the mirtron technology is a more, I'll call it smart platform,
Mm-hmm.
but smart, as it's smart, it requires also computer tools, ability to predict. Even the mirtron itself, the design of the mirtron needs to be a targeted sequence. So to select that sequence correctly, so the effect will be at the right level. So there are a lot of parameters in the mirtron technology that requires AI. So that's one of the examples where you develop something that now maintain the plant physiology intact, keep the yield intact, all the parameters safe. You want to just now have nematode resistance at the root. That's
Mm-hmm.
all you want. And then you can do that using AI tools combined with our platform.
Wow. Thank you so much, Kinneret, for taking us on a journey into your technology and into your mission. And I can tell you have a lot of passion behind both of them. I hope you will be back maybe after that the project that you teased that is in the making has come to fruition and is in the market. For now, I invite our audience to check out our show notes. We will, of course, be linking to GeneNeer, and please check out Kinneret's work to learn a little more. Thank you for being here.
Thank you, Anna. Have a good day.
Thank you for listening in today. Want to learn more? Then check out our show notes and more info on computomics.com. If you have questions or want to propose a next guest, please reach out to us at podcast@computomics.com.